Hello med students. In this video, we will discuss CD4 positive T-cell differentiation and function. By the end of this video, you will be able to look at this crazy diagram and explain the general functions of CD4 positive T-cells, understand the process of T-cell maturation through positive and negative selection, and know the 4 different subtypes of these CD4 positive T-cells, as well as their cytokine feedback loops. First, let's give ourselves some context and look at the immune system, which is made up of 2 broad parts, adaptive and innate. The adaptive is made up of T cells and B cells that use antigen-specific receptors. Within T cells, there exist 2 types of cells called CD4 positive and CD8 positive T cells, which are each defined by which of 2 receptors they have, CD4 or CD8. CD4 positive T cells bind MHC class II and are called helper T cells because they primarily function to activate other cells, including B lymphocytes, macrophages, and dendritic cells. Now let's begin our overview of T cell maturation. The importance of T cell maturation is having the right membrane receptors, and we'll see how our cells get trained by our body to have the correct receptors through positive and negative selection. Initially, T-cell precursors leave the bone marrow with neither CD4 nor CD8. This is referred to as a double negative. They travel to the thymus where they will develop to double positive, which means T-cells with both CD4 and CD8 present. The thymus is comprised of an outside cortex and an inside medulla. Positive selection takes place in the cortex and proceeds to negative selection in the medulla. Let's dive into what these processes are. The term positive selection really means that those that bind will be selected to survive. In the cortex, positive selection allows for T cells that express CD4 or CD8 to move on to the next step. If T cells recognize MHC1 or MHC2 molecules expressed in the cortical epithelial cells, then they will survive positive selection and will travel to the medulla to undergo negative selection. T cells with CD4 that recognize MHC class II will remove their CD8 receptors, while those with CD8 that recognize class I will remove their CD4 receptors, rendering the T cells either CD4 or CD8 expressing T cells. They are no longer double positive. If the T cells are unable to engage with either MHC class expressed on the cortical epithelial cells, then it will not be supported metabolically. This neglect causes the cell to die via apoptosis. Remember what I said, positive selection is selecting for those that are CD4 or CD8 positive. In the medulla, we enter negative selection, which means that those that don't bind are selected to move on. In this second stage, we are developing central tolerance, which means our T cells should tolerate self-antigens and not elicit an autoimmune response. If a T cell recognizes MHC1 or 2 molecules presenting self-antigen expressed in the medullary epithelial cells, then they will be induced to apoptose via signal 1, which is called TCR-induced cell death. This is filtering out T cells that would otherwise cause an immune reaction against our own body. If the T cell is unable to interact with MHC1 or 2 presenting a self-peptide, then it is not recognized as autoreactive and permitted to be part of the matured T cell pool. Once the cell leaves the thymus and enters the lymph nodes, it differentiates into one of 4 different subtypes of CD4 positive cells, each of which has unique cytokine activators, inhibitors, and functions. These subtypes are called Th1, Th2, Th17, and Treg. We'll start with Th1. Th1 cell differentiation is driven mainly by cytokines interleukin-12 and interferon gamma produced by the innate immune system cells such as dendritic cells and macrophages in response to a microbial infection. Once Th1 cells develop, they secrete interferon gamma, which promotes more Th1 differentiation, amplifying the reaction. Interferon gamma production also inhibits differentiation of Th2 T cell subsets, promoting polarization in only one direction. When Th1 cells are stimulated by antigen, they express CD40L on their cell surface. Th1 cells can then activate macrophages via binding of their CD40L onto the CD40 receptor of macrophages. Activated macrophages have enhanced killing of phagocytosed bacteria. Th2 cell differentiation is stimulated by interleukin-4 in response to helminths, parasites, and allergens. Like Th1, a positive feedback from activated Th2 cells produce interleukin-4 to induce further Th2 differentiation. Activated Th2 cells also produce interleukins 5 and 13. Like Th1 cells, Th2 cells also exhibit a CD40L, but Th2 instead binds to the CD40 receptor on B lymphocytes rather than macrophages. This allows for B lymphocyte antibody class switching. In this case of a parasitic helminth infection, our Th2's CD40L binding and IL-4 production stimulate B cells to class switch and generate IgE antibodies, which aid in fighting parasitic infections with helminths. For our third CD4 positive subtype, we'll briefly discuss Th17 cells. Th17 cell differentiation is stimulated by pro-inflammatory cytokines, including IL-1, IL-6, and TGF-beta, which are produced in response to bacteria and fungi. Activated Th17 cells produce IL-17, IL-21, and IL-22, which are inflammatory cytokines important for neutrophilic inflammatory response. Remember, Th17 neutrophils. The last subtype of CD4 positive cells we'll discuss are regulatory T cells, or just Treg. Treg are generated by self-antigen recognition via cytokines IL-2 and TGF-beta, and these function to suppress immune responses. Tregs produce IL-10 and TGF-beta, which are anti-inflammatory cytokines. Tregs are able to suppress CD4 positive and CD8 positive T cell effector functions, thus regulating the T cell activity. CD4 T cells are an essential part of adaptive immunity, serving to activate other cells through cytokine pathways. Through positive and negative selection, it is ensured that CD4 cells protect our body from invaders without attacking our own cells. The different types of CD4 cells are summarized in this flowchart.